So hello students, you have already studied about healthy cell back in your high school as well as in the histology. You know everything about the normal cell, how it functions and everything like its biochemistry, the cellular physiology and all. About the cellular adaptation, we have already discussed in our previous lecture like hypertrophy, hyperplasia, metaplasia and atrophy. While in this section, we will be discussing about the reversible cell damage and irreversible cell damage. So this was the template which we had followed. This is a healthy cell. When we put stress on the healthy cell, it tries to adapt. It tries to adapt by hypertrophy, hyperplasia or metaplasia. But once the adaptive capability of cell increases up to a limit, the cell undergoes damage. This kind of damage is known as reversible cell damage, right? Reversible cell damage means if we remove the stress, the cell can go back to the cellular adaptation stage. And if we again remove the stress, the cell can go back from cellular adaptation to healthy stage. But if we do not remove the stress and if we let the stress continue, the reversible cell damage will convert into irreversible cell damage. So the point is, there is a healthy cell, we put stress, cell tries to adapt, but if the stress increases the adaptive capability of the cell undergoes reversible cell damage. If we do not remove the stress and let the stress continue, the reversible cell damage converts into irreversible cell damage. So today's lecture will be reversible and irreversible cell damage, and in the next lecture we will see if we do not remove the stress even after irreversible cell damage, the cell will eventually die. Okay, reversible cell damage. Well, what is the most common cause of cell death? Do you know? The most common cause is hypoxia. Hypoxia means low oxygen at tissue level. What is the most common cause of hypoxia? That is ischemia. What is ischemia? Low oxygen delivery by the blood through blood vessel. Suppose this is your artery. This is carrying blood as usual. But what happened is atheromatous plaque came here. And now when the blood tries to, go across this blood vessel, this plaque will prevent adequate amount of blood to go to the tissue. So we have reduced the blood supply to the tissue, and this is ischemia, low oxygen delivery by the blood vessels. Got it? So this is ischemia, which is lower oxygen delivery by the blood through the blood vessel. Why? Because there is a obstruction in the blood vessel, while the hypoxia was low oxygen concentration at the level of tissue. Now which is the most sensitive cell for the hypoxia. Most hypoxic sensitive cell is our neurons. And which is the most sensitive cell? So which is the most resistant cell for hypoxia? That is our fibroblast. Most sensitive is our neurons. Right? So the most common cause of the cell death is hypoxia. What is the most common cause of hypoxia? It is ischemia. What is the most sensitive cell to oxygen? That is our neurons. And which is the most resistant cell to hypoxia? That is our fibroblast. Got it? Now we will be studying our reversible cell injury, taking hypoxia as a cause. Even if you read the Robbins, they have also taken hypoxia as a cause of reversible cell injury. Well, there can be other cause, but most common cause is hypoxia. So first of all, before we start all the stages in the reversible cell damage, you will see there will be three things. What are the three things which will be there? Cell membrane will be damaged. In the cytoplasm, you will also see a change which is reduced protein synthesis, and in the nuclei you will see chromatin condensation. So this is what we will be seeing in the reversible cell damage. All the three essential components of the cell, which is the cell membrane, it will be damaged, it will show blebbing. The cytoplasm will have reduced protein synthesis, and the nuclei will show the chromatin condensation. So let's begin. Suppose this is hypoxia. Hypoxia means lower oxygen at the level of tissue. If there is low oxygen, which organ will be affected first? That will be our mitochondria. Why? Because mitochondria is our powerhouse of the cell. It produces ATP using the oxygen. So it will be affected first. So if someone asks you which organ is the first affected organ in cell damage, you have to answer it's mitochondria. Now, if there is no oxygen and the mitochondria is affected, what will happen? Cell will undergo anaerobic respiration. Now, in anaerobic respiration, what is the result? At the end of anaerobic respiration, you get a few ATP and you get lactic acid or pyruvic acid, also known as lactate or pyruvate. What do? They will reduce the pH. When the pH reduces, it means this is acidic environment. What does acidic environment do? It causes clumping of nuclear chromatin. Did I say in the reversible cell damage there will be three things? Nuclei will be affected. So yes, first thing we have seen the chromatin condensation happening here. Now, since the mitochondria is affected, mitochondria produces ATP. And if the mitochondria is not producing ATP because it has undergone failure, less ATP production. If there is no adequate ATP, sodium potassium pump won't work properly. What is the function of sodium potassium pump? Its function is, let's say this is a cell, its function is to keep potassium inside and sodium outside. But if this pump is not working, what will happen? Sodium will start entering inside. If the sodium has started entering inside, what will happen? Osmolarity of cytoplasm will increase. What happens after the osmolarity of cytoplasm increases? It attracts more water. Once it has attracted more water, what will happen? Cell swells. Now, let's take a break. Try to understand it this way. You go to kitchen, you take a tablespoon of salt and you put it on the counter of your kitchen. What happens is after 5 or 10 minutes, you will see earlier the salt was dry. Now the salt has attracted water because every solute in its purest form has highest osmolarity. So here we also saw that the osmolarity of the cytoplasm increased. It means it can attract more water. So since the sodium entered inside, the osmolarity of the cytoplasm increases, cause of which it attracted more water, and if there is more water inside the cell, cell will swell. Can we say cell swelling as ballowing? How we blow the balloon. So this feature is known as ballowing degeneration, or since the water is coming inside, this is known as hydropic degeneration. Now tell me one thing, since the sodium has come inside the cytoplasm, will the sodium only stay inside the cytoplasm or it will enter inside the endoplasmic reticulum and other organelles also? The sodium will enter inside the organelles also. So they will also swell, right? Your endoplasmic reticulum will swell. Now, if the endoplasmic reticulum is swelling, what will happen? Normally, endoplasmic reticulum is attached onto the surface. Now, since this has swollen, they will fall off. What will happen? ER swelling. What is the result of ER swelling? Ribosomes fall off. Now, what is the function of ribosome? Synthesis of protein. Now the ribosome has separated from the endoplasmic reticulum. Two things will happen. First of all, protein synthesis will be decreased. And what is the second thing that will happen? Now, RER has converted into SER. What is the function of SER? Synthesis of lipids. So this is the reason why when an organ is not working properly, let's say your liver, they start producing fats or the lipids, and we call it what? Fatty liver. So always remember it. So as a result, what happened? We saw protein synthesis is disrupted. Did I say this will be another thing? Right? Now the third thing, the cell has swollen from this much to this much. Now tell me one thing, if this is a cell, and let's say this has microvilli. Now the cell has swollen. Those protoplasmic modification will be lost, right? So as a result of the cell swelling, loss of protoplasmic modifications occur. Got it? Yes. Now still the cell is swelling. What will happen? If this was a plasma membrane, there will be a part which will undergo weakening, and in this part a bleb will form. This is known as membrane blebbing. This is a weaker part in the cell membrane which has undergone damage. And did I say the cell membrane will undergo damage? So, reversible cell injury, most common cause is hypoxia. We saw how hypoxia caused the cell membrane damage or the membrane blebbing, how it caused cytoplasm, reduced protein synthesis, And in the nuclei, we also saw how chromatin condensation took place. Let me show you some photograph. Now, this is a photograph of your PCT of nephron. What is the general histology here? Breast border, cuboidal epithelium. It means it will be cube-like cell with the nuclei in the center, and it will have breast border or the microvilli. Can you see? This is a cuboidal epithelium. What does it have on the surface? It has breast border, but here it's reversible cell damage, right? Can you see now these microvilli? Or you see that the cell is swelling? You see small vacuoles here, which are probably the lipids, because I said there will be fatty degradation or fatty degeneration, or lipid synthesis will increase, and you see these microvilli are finally lost here. So I think you got it now. At the end of reversible cell damage, we will see the damage of the cell membrane in form of the membrane blebbing. How does membrane bleb appear? They appear in this way. So this is our normal cell, and I said after the hydropic degeneration has taken place, small blebs will occur in the cell membrane. You see this small spherical outgrowth, these are the membrane blebs. And one more thing, since this is the weak part, some amount of phospholipid will go into the cytoplasm, and this will mix in with certain amount of calcium, and this is what we give it a name known as myelin figure. Why do we call it myelin figure? Because when you had studied nerve, if this was axon, myelination takes place in the form of hold. Similarly, you see this myelin figure, this one or this one, this is like this vortex like pattern. So this is the reason why we call those phospholipid calcium composites as myelin figure. In the electron microscopy, they appear as black and white. This is the notes which I will be giving you guys. This is how it appears in the electron microscopy. Got it? So this was our reversible cell damage. Now for the irreversible cell damage, everything will remain same. The protein synthesis will be reduced, but very much. The membrane, the cell membrane will be damaged, but a lot. The mitochondrial change will take place, but very badly. So irreversible cell damage. So in irreversible cell damage, we will see two things primarily: severe mitochondrial damage and severe membrane damage. 2MD, right? There is severe membrane damage. What will happen? Earlier only sodium could come inside, because now the sodium potassium pump wasn't working. So sodium started coming inside. But now there is membrane damage. There is severe membrane damage. What will happen? Severe ion influx. All the ions which are supposed to stay outside of the cell, for example, even more sodium, even more potassium will come inside, even more calcium will come inside, even more magnesium will come inside. Normally, let's say for magnesium is, like it helps in the translation, it binds with the smaller subunit, so it helps in that. Calcium, on the other hand, this is very dangerous for the cell, because normally only 1% of the calcium stays inside the cell, while the rest stays outside. Why? Because calcium activates enzymes. And once the enzymes are activated, what will happen? Let's see. Protease. It will cause the destruction or the denaturation of protein lipases or lipase. What will it do? Phospholipase will cause phospholipid breakdown. Similarly, we will have nuclease. All of these enzymes are already present inside your cytoplasm, but they are present in inactive condition. But when the calcium enters inside, they gets activated. So nuclease will also cause the destruction of your nucleic acid, but it doesn't happen in a single stream. First of all, there will be nuclear chromatin condensation, known as pycnosis. Then the chromatin will break into fragments. This is known as karyorrhexis. Soon these fragments will completely be dissolved, known as nuclear dissolution. This is known as karyolysis. Karyo means nuclear, lysis means breakdown. So this is how it will appear. Now you see, the pycnosis, the nuclear chromatin is undergoing condensation. If condensation means what? Increasing something. So if this was the chromatin, this has now condensed. So earlier, if it was light blue, now in the microscopy, what will it appear? It will appear as dark blue. And in the cell, cytoplasm appears as pink and the nuclei appears as blue. So in the beginning, it will be light blue, but when there is irreversible cell damage, this will go even darker. This is pycnosis. Soon they will break into fragments, and soon these fragments will even dissolve, and only pink colored cells will be left. And this is what you see as a result of irreversible cell damage, and the soon cell will die. Got it? So we saw how there was membrane damage. Membrane damage caused the influx of calcium. Influx of calcium activated enzymes. Activation of enzymes like proteases broke protein. Phospholipases broke phospholipid. Phospholipid is the composition of the cell membrane. If the cell membrane is now broken, more calcium will come inside, more breakdown. So this is irreversible, because now you cannot go back. This is like a domino. This will only proceed further. Similarly, you had nuclease which caused pycnosis, karyotaxis and the karyolysis. Now I told you the ions won't enter just inside the cytoplasm. It will enter inside your mitochondria also. So when this calcium entered the mitochondria, what will happen? Mitochondria is already not working because it didn't had oxygen. So what will happen? It was already mitochondrial damage. Now the calcium entered inside the mitochondria. Now the calcium, it will activate even more phospholipases. That will also destroy the phospholipids of the mitochondrial membrane. Those phospholipids will associate with calcium ion and they will form what? Amorphous densities, also known as amorphous flocculent density. Got it? Amorphous flocculent density inside the mitochondria is a sign of irreversible cell damage. Now I think you guys understood. What was the first sign of reversible cell damage? Hydropic degeneration. What was the sign of, not the first sign, what was the sign of irreversible cell damage? Severe mitochondrial damage, severe membrane damage. Because see, severe membrane damage, it means earlier it was 5 blebs, now this has gone 500. So even in the reversible cell damage we had blebbing, but in the irreversible the blebbing number will increase. So this was our reversible and the irreversible cell damage, and as a result of this, cell will proceed into cell death. Got it? And we will see this kind of cell death. Normally the cell death occurs in two ways. One is necrosis and another is apoptosis. While the apoptosis is genetically programmed, necrosis is pathological, and we have seen there is a pathology involved here. There is a disease which is causing it. And here we will study necrosis, and the necrosis in the later class will be coagulative, liquefactive, fibrinoid, caseating and all those. So for now, this is all. We will see it in next class. Have a good day.